So this is a large research team, and in fact, our first paper in Science had 31 co-authors. There's about 50 people up there, if you count them. Kelly was actually out in the field collecting samples too, herself. She's not in the picture because that's all the people that were at the lab at the time. And as we said, this is supported by EBI. We have a number of other collaborators too. There is, let's just go through some of the scenario of what happened. On April 20th, the Deepwater Horizon oil rig, which is actually a vessel, exploded for unknown reasons, but it was probably rising, rapidly expanding methane. The floating rig burned for 2 days, killing 11 people, and then sank, shearing the 21-inch riser pipe at least 600 feet above the blowout prevention device. There were 3 breaks in the riser pipe, and this originally the extent of the oil was placed at 5,000 barrels a day. That was quickly revised, which there was a lot of criticism about, to up to 60,000 barrels per day. 54.1 million cubic feet of natural gas was flared, and totally we believe now that about 4.9 million barrels of oil was released during the spill. However, BP has a lower number that they think is probably more appropriate. Oil and natural gas, probably. And I'll show you the breakdown of that in a minute. The spill has surpassed the Exxon Valdez spill, which was the second largest spill in the United States. It also surpassed the Ixtoc 1 blowout, which was 3.3 million barrels in 1979. Ixtoc 1 blowout was actually the second largest marine spill in the world. The first is still the Gulf War oil spill, and where it was more than 9 million barrels of oil went into the Gulf of Arabia, especially oiled all the marine areas along Saudi Arabia. And we can still find that oil yet today. The well was capped beginning July 12th, and no more oil went into the Gulf of Mexico after July 15th. Now you'll see from the Macondo well, and the Macondo well was declared dead on September 19th. Now you're going to see 3 different names. Deepwater Horizon people use, MC252, which is Mississippi Canyon 252, which was the parcel where that well was drilled. The name of the well is Macondo. All of those things are the same thing. And so you'll see them used interchangeably. Now, this is what happened. You can see the oil at the surface, and then you can see it, the breaks in the riser pipe. And of course, the depth here is over 5,000 feet, 1,500 meters. And there was 3 breaks in this, and the pipe coiled around as it was dropped from the ship. They put in a relief well, started drilling a relief well almost immediately, got their ROVs in there, did a top hat, and that failed. Subsequently did here the top hat, and they tried various other containment things, and then they also went in and tried to do a siphon. First time that failed, but the second time they were able to insert it into the riser pipe and then started pumping some of the oil to the surface. They started a second relief well to make sure they could shut it off. And then they started doing various other operations of a top kill and injecting junk and drilling mud and a variety of other things into the blowout prevention device to try to kill it. That didn't work either. They finally cut off the riser and put a cap on it and tested that for a couple of days, and on July 15th, the oil subsequently stopped completely. And you can see the progression of the oil at the surface here, and this is all from NOAA. And then here it's capped and stopped, okay? Now, the oil is, from the Macondo well, is a typical Louisiana light crude, and that's quite significant because this oil is inherently biodegradable, and remember, oil is basically algae and plant material that has been exposed to extreme temperatures and pressure. And this particular oil, because it's so deep, has been degraded even more and has a very large number of light components. And the reason why the oil companies are going quite deep on this is because it does have these light components, which are basically BTEX types compounds and fuel compounds, quite important, but a low amount of these high molecular weight compounds. In comparison, here is the Exxon Valdez tanker spill oil, and you can see it's much higher amounts of these higher molecular weight compounds going out to 32. And then this particular oil has been very well characterized. It's typical of these reservoirs that are over 6,000 meters. We know the isotopic signature, carbon isotopic signature. About 35% of it will evaporate in 2 days, very high volatile component, 45% in 2 weeks. Now, the oil is quite different than if it was a tanker spill and it came out over the surface and it was heavy oil. This is light oil, and it's being jetted into the deep subsurface in the marine environment and literally jetted at a very high pressure. And it causes it to form droplets when it mixes with the water, and especially with the temperature differentials and that sort of thing. Remember, the temperature at this depth is about 4 degrees. Maximum density water. So all of these lower depths are below 5 degrees. If the droplets are about 10 to 60 microns, they have a neutral buoyancy, and basically they can move out with any sort of current that occurs. And there was a current from 900 to about 1,300 meters, and we did see this deep water plume, and this is the first time we've documented this before. Larger droplets move up to the surface, and then there was skimming operations, burning operations, because this is highly volatile, and they were adding dispersant to try to keep it from coming to shore. Now remember, it was almost 50 to 100 miles from shore, so quite far from shore. But they did put booms down, that sort of thing. Occasionally, there was material that got to shore and it formed tar balls and that sort of thing. There was also a lot of methane in this and hydrocarbons here, and some of that moved out with the current. As it went to the surface, it formed tar balls after it emulsified and that sort of thing, and I'll talk about that in a minute. Now, for the first time, EPA gave approval for BP to inject Corexit 9500, which is a common dispersant, at the wellhead. Now, the primary reason that BP did that, and there's just all sorts of wild speculation in the media, but the primary reason that they did it is because they were there was over 7,000 ships out here at one time. And a lot of these ships were concentrated around the wellhead trying to close the leak. If all of that oil had moved directly to the surface, which it did initially, it was going to be a safety problem in terms of explosion and flammability and that sort of thing. The Corexit created some smaller droplets and actually caused the oil to move farther out. Did it stop it from moving to the surface? No. But within 4 hours, it was moving farther away and made it safe for the operations around the leak operations around the wellhead. And every time they shut the Corexit off at the wellhead, when they're doing various operations, 4 hours later, more of the oil was moving to the surface. So that was well established. Again, 4.9 million barrels, 3% skimmed, 95% burned, and these are the latest statistics from the December government report. Chemically dispersed 16%, naturally dispersed 13%, captured 17%, 23% evaporated or dissolved, and 23% remaining. That's probably, a lot of that has been biodegraded now, except that went to shore. 31,800 people were involved. 1.84 million gallons of dispersant were used, largest amount ever used. 441 controlled burns, and 3,447 kilometers of containment, boom, and absorbent. They cornered the market on containment, boom, and absorbent. In fact, there was no more sources anywhere in the world. They had used them all up. I'm not sure that was the best strategy. In fact, skimming and operations and that sort of thing might have done a little bit better. So what we were trying to do was, like we're doing with the Microbial Enhanced Hydrocarbon Recovery Program, is apply sort of oil systems biology, looking at geochemical processes, microbial population correlates to geochemistry, measurement of biomolecular activities during the process, simplified laboratory communities, cellular network analysis of function, molecular analysis, and then doing predictive models of cellular function, predictive models of community function, and predictive models of geochemistry and ecology. To do that, we were looking at microbial community structure and function with in situ analyses with the Berkeley phylochip, geochip, phospholipid, and proteogenomics, metagenomics, metatranscriptomics with JGI, doing DNA and RNA sequencing and functional structure. And then we were also isolating various organisms in lab simulations for physiological characterization and rate determinations, the rates of biodegradation of the oil itself. We used a synchrotron advanced light source for FTIR real-time analysis, high pressure simulations, and we used the microrespirometer for rates of biodegradation under as many as 30 conditions, simultaneously monitored in the real time. We also used phenotypic microarray for rapid biochemical characterization of the isolates and biodegradation rates, and minimum inhibitory concentrations of the corrects, et cetera. We also did a lot of geochemistry for linking structure and function using systems biology approach, so stable isotopic analysis and of both the petroleum and daughter products, and then linking the biogeochemistry to petroleum bioavailability and community populations, and then trace metals and limiting nutrient characterization. We were on 4 different ships from May 25th to October 20th. And so unlike a lot of the other scientists that were out there, were usually they were out there for no more than a couple of weeks, we were out there continuously from May 25th to October 20th. Now, of course, we came in for shipping samples back and that sort of thing, but for that whole time, we had people down there. And we did various missions. We filtered samples for ecogenomics, 1 to 20 liters, and BP had ultra cold freezers brought on board the ships, which is one of the first times I've ever seen that allowed us to immediately flash freeze the samples in the filters. We had fixed samples for direct counts in hydrocarbons, isotopes, metals, and nutrient analysis. And our laboratory is a certified EPA laboratory for doing that type of analysis. We also had live samples for isolation and lab simulations. We went from 1.2 kilometers to 384 kilometers in all directions from the wellhead. Collaborations with the University of Alabama, Florida State, Gulf Coast Research Lab, Rutgers University, University of Oklahoma, Penn State University, University of Georgia, Scripps, and University of Tulsa. If you go to this particular web page and wiki, you can see all of the ships that we were on, and a lot of the data, the papers, and even the daily reports. And that's been up there since the day we went out. So publicly, everything's transparent here. This is at the wellhead, and they're flaring the gas that they were bringing up. And you can see that over here too. This is a controlled burn. Sometimes the oil would look like this. 99% of the time, the oil was looking like this in terms of a sheen at the surface, a very light sheen. Occasionally, we would see this red mousse. This is the oil mousse. And this red color is related to some of the pigments that were originally in those plants that were involved or algae that were involved. We also took, while we were on shore, we occasionally took samples for sediment, and this is at Elmer Island, and they have a separate study that we have ongoing on that. For surface samples, we used a Halliburton bucket, which was appropriate probably. And it's interesting, this is in a Odyssey magazine, did a special, and this is the K through 12 magazine for Adventures in Science for kids. And I sent them this picture, and they photoshopped out Halliburton on this. It was interesting. So anyway, you can see here a collection from the CTD. So these could be lowered in the water. They had a number of different instruments on them. We could measure DO, conductivity, temperature, pH, pressure, and then also there was 2 fluorometers on there so that with the fluorometer we could detect oil. And then on the trip back up, we'd go all the way to the bottom, and on the trip back up we would then fire the bottles at a particular depth to collect the samples. And we'd take samples within the plume and above it and below it, and then at the surface, of course. And this, of course, is some surface samples, heavily oiled, and we were filtering those in the lab. And the lab being on the ship. Again, here's taking samples with the CTD, which stands for conductivity, temperature, depth. And this shows a typical CTD profile on the raw data. And here you can see the fluorimeter and see this spike at around 1,200 meters. And this shows that there's oil down here. And then we can also see this dissolved oxygen curve and see that there's a decrease in dissolved oxygen occurring at almost exactly the same place. There was some concern that the instruments were being affected by the oil, but it's very low concentration of oil. We actually, per BP, we did standard Winkler assays and verified that in fact the instruments were right and correct. In fact, we're not being affected at this concentration. The concentration of this, the highest concentration we ever saw at depth was 10 parts per million, okay? So it was low concentration. And actually you could not see it or smell it in those deep samples. And then we have also pressure and that sort of thing too. Camilli and his group at Woods Hole did a bunch of instruments, took a bunch of instruments out there and published about 4 days before us in Science the deep plume bathymetry. Now, they showed it was going out to the southwest. This is the wellhead here. And they saw it in exactly the same place that we did. They actually, Science gave us their paper so that we could cross-reference to it, and subsequently Camilli has cross-referenced to ours. They made some speculations that the oil would not go away that fast based upon some old literature values, though they did not take any microbiological analyses like we did. The blue line is how they traverse their instruments across this area, and the black area is their extrapolation on concentration of where that oil was, okay? And it's following the bathymetry along this canyon, deep canyon, to the southwest. Our samples initially on the science paper were taken here, and this is the wellhead. And we were on 2 different ships, the Ocean Veritas and the Brooks McCall. And subsequently, you were on the Brooks McCall, weren't you? And then subsequently, did you go on the Farrell? Yeah. And then you can see we have both plume detection and that sort of thing, but most of these samples where we detected oil were out to the southwest. We, however, did detect one up here to the northeast or northwest, and there was a couple anomalies like this. This was probably due to the tremendous amount of oil that was coming out of the riser until they cut it off, and that all of the oil was coming directly from this one source. But the risers were coiled around here, and remember there's about a mile of riser that collapsed onto the sea bottom. So the concentrations of alkanes, and 80% of this oil is alkanes. So a large proportion of it is alkanes. And that was the easiest thing for us to measure. We could see it's stratified here at about 1,300 meters or so. And this whole interval from about 1,300 meters to about 900 meters, we saw oil in. Here is the BTEX compounds, benzene, toluene, ethylbenzene, and xylene. And you can see it's up a little bit higher. And these, of course, are more volatile. We didn't see any of that at the surface. And the PAH compounds, very low concentrations of those, but they're very volatile. And almost all of that was detected only at the surface. We didn't detect any down deep. Now, basic oil biodegradation. So the simplest way to say this is that the petroleum the bugs provided enough, if they're provided enough oxygen and the correct nutrients in terms of nitrogen and phosphorus, will degrade this material they can degrade it completely to carbon dioxide and water, but they also can degrade it to other daughter products. And there may be only a certain set of enzymes that they have, so they can only go to particular daughter products, one particular bug. But in a community, they can totally degrade everything that's there. What happens at the down deep is we see biodegradation by pelagic organisms. We see biodegradation in the benthic regions and sedimentation. We also see some absorption to particulate matter, dissolution, and sort of a cease oil slick. And then, of course, evaporation, photooxidation is occurring, and then drift and spreading, emulsification, and the formation of the mousse. This is actually the most difficult thing to biodegrade. And so once this got to shore, it's probably going to stick around for a lot longer, as we'll see. So what we saw was that at 1 kilometer, we actually see very little change in total bacterial counts, but a very strong signal for the oil down deep. But at 5 kilometers, the bacteria densities have more than doubled and the oil has decreased slightly. And then out at 10 kilometers, it's decreased a lot, and we still see a very large bacterial abundance. When we go outside of the plume, and this was only 5 kilometers away, but was at an area where we didn't see any oil at all. We don't see any difference in the bacterial densities. Down deep, also with looking at dissolved oxygen, initially we don't see much of a dip in the dissolved oxygen, just very slight. But at 5 kilometers, it was quite, and this is following the current, at 5 kilometers, it was quite significant. Once the oil, a lot of the oil has been degraded, there's less dissolved oxygen being consumed. So out at 10 kilometers, a little bit less. And we don't see any of this difference out in the non-plume areas. Statistically, phosphate levels were significantly lower, and I keep forgetting to label this doggone graph. So these are the plume samples, and this is the non-plume. And phosphate levels were significantly lower, but not significantly. The ammonia levels were higher, but not significantly. The nitrate levels were significantly lower, as you would expect if they're in fact sucking this up. The dissolved inorganic carbon C13 ratios were not significantly different. That's probably mostly due to the fact that the methane wasn't being degraded at this point. As we'll see later, it was later. Other hydrocarbons were dramatically different in terms of concentration. Biological density was significantly higher. Archaeal richness was not significantly different. Now, the archaea would be a lot of the methanogens, okay, and those types of organisms, and anaerobes. No difference there. This was a completely oxygenated environment. Bacterial richness was significantly lower, as was total richness. And this is, as we'll see, this was due to the fact that the oil degrading species came up, but everything else dropped back, okay? And the ocean of Spirales, which was one of the dominant clones, was significantly different. Phospholipid fatty acid analysis in terms of total lipids also showed that there was a significant biomass difference. The cis-trans ratio of the lipids in the membrane, which normally would indicate some sort of stress, showed that there was no significant difference. And then the ratio of certain lipids that we know are associated with oil degradation were significantly higher also in the membrane. So again, multiple points of evidence. And something that's this controversial and dramatic, you have to have multiple sources of evidence for everything. This just gives you a more complete picture of the lipids. In terms of biomass, dramatic difference in biomass between the plume and non-plume. Also, we can see a difference in community structure with the lipids, and this is the average plume versus the average non-plume. And there's we can go through all of this, but also, we'll go through some of the 16S. We use the Berkeley Lab phylochip, which has 50,000 different bacteria and archaea in one test. And the location and intensity indicate the amount of that particular DNA that would be found for a particular OTU, for example. And it's 16S. We found 951 subfamilies that we detected in 62 bacterial phyla. Only 16 subfamilies in the gamma proteobacteria were significantly enriched in the plume. So we could see this from the phylochip. So here you see the gamma proteobacteria enrichment. And by the way, you can pass oil around while I'm talking. This is the Macondo oil, and you can see how light that is. It's more of a brown color. This is weathered Macondo oil. And don't drop this because this is more valuable than platinum, I think, right now, because the well's dead. But you can see when it's weathered, it's completely different characteristics. And so the gamma proteobacteria, we had about these 16 subfamilies, Ocean of Spirales, the Coeliaceae, and all of them have either the ability to degrade hydrocarbons, or we know that they can be enriched by crude oil. And we can go to that from particular species that are known to occur in these particular classes, families and subfamilies, and go to the literature on this. So one of the amazing things is how well the lipids compared to the 16S rRNA. So the ribosomal RNA, actually, for all the samples that we took that were within the plume versus non-plume, actually gave us the same sort of structure. And this is using a multidimensional scaling ordination of Bray-Curtis distances. And we did some stress tests on this and that sort of thing. But basically, we can show that there's very highly significant differences between the clusters here. And they're remarkably similar in terms of plume versus non-plume. So again, completely different methods of analysis showing that there's an extreme difference between the bacteria community that's in the plume and outside the plume. We also did clone libraries and did 200 or 300 of these. And when we looked at a particular well or particular sample where it was non-plume sample, Only 5% of the clones that we found were the ocean aspergilles clone that we saw. Whereas within the plume, it was either 90% or 95% of the clones. So this was one of the dominant subfamilies and bugs in there. And of course, that is a psychrophile, and it will degrade oil. We know, and a psychrophile means that it will grow better at 4 degrees than it will at 20 degrees. And we'll see that that was important in terms of the oil too. We also did rarefaction curves on our microbial diversity. And you can see here from the non-plume samples versus the 2 samples that were within the plume that we did metagenomics on and sequencing efforts on, and how different those were. Again, microbial community composition, and this is from pyrotag sequencing of 16S, and you can see the 2 plume samples here, and then the non-plume samples, how different the diversity is here. It's very clear. Not being satisfied with that, we also did the GeoChip, which is a functional gene array. It has a number of different genes on it that we believe are highly conserved for particular functions like methane metabolism, nitrogen cycling, contaminant degradation, and carbon degradation. So specifically oil degradation. And we applied the DNA that we extracted to that. And you can see here for all of these particular classes of cycloalkanes and alkanes and alkynes degradation genes, they were all significantly higher in the plume than they were outside the plume. Also, the aromatic carboxylic and acid degradation genes were also all significantly higher in the plume than they were outside the plume. We also went and did a direct correlation between the different oil components and their concentration and these particular genes that we found. And again, anything here in bold, if you can see that, indicates that there was a direct correlation between how the concentration of that particular gene or the amount of gene that was being expressed and that particular compound that it could degrade. So again, nice correlation there. For the direct counts, this was actually showing at 1 kilometer out, and you can see that the bugs have already started to divide. And if you look real close here, it's easier to see on the science paper. You can see that there's some septa in here, so they've not completely divided, so it looks like they're 20 microns long, but there's actually incomplete division here. So actually, I was counting all these as one cell. So I do all the counts myself, by the way, okay? And I was counting this as 1 cell and this is 1 cell rather than try to go down there. So I probably underestimated the total number of bacteria that were at 1 kilometer even. And so it would only, because the current was moving at 7.8 centimeters per second, at that depth, they didn't have very long to start responding, and they responded very, very rapidly. Out at 10 kilometers, they're fat and happy, they've completely divided, and you can see there's much higher densities. We took live water samples back to the lab. And the standard way that you take live water samples back is you refrigerate them at 4 degrees. Well, that didn't work out real well, okay? Because these bugs were adapted to growing and doing well at 4 degrees, when we got back to lab, sometimes it was only 48 hours. When we analyzed them, there was no oil there. They had completely degraded it, and what was left was these flocks, okay? So this is sort of bacterial poop or exopolysaccharide and a variety of other materials. So using the synchrotron FTIR, we could see that there was proteins and carbohydrates associated with bacteria. And we could also see the very low amounts of alkanes were still present in the flocks. Now these are practically undetectable by any other means. But also lots of oxidation products, lots of nitration products and sulfation products, indicating they were very, very active against degrading the oil. We did take oil from the surface, much larger droplets, and as you can see here with this heat map for the alkanes, this is a large oil droplet. And we could see bacteria attached to the oil droplet, and again we can see with the protein heat map that this is a bacteria. And then we could see the oxidation products associated with that bacteria that we know will degrade the oil, so they were attacking it directly. So when we brought that back at 4 degrees, it did preserve them at the surface, because the surface temperature during the summer there was over 30 degrees C at the water surface most of the time. And here you can see, I hope you can see this. This is better in in the science paper and some of this, but you can see the bacteria actually attached to the oil droplet here and here, and the oil droplet, as you can see here. As I said, hydrocarbon biodegradation pathways to degrade compounds found in crude oil by marine microbes include everything. And they can degrade virtually all components, even though crude oil is hundreds and hundreds of different compounds. And they can degrade it completely to CO2 and water. So, what sort of biodegradation rates did we get? What we did was to look at estimating conservatively how fast the oil would move in the plume, and it would either go out, it would take 2 days to go out 10 kilometers or 5 days. We took and analyzed that for all the alkanes, and then looked at how fast they could degrade. Considering the changes that occurred from 1 kilometer out to 10 kilometers. BP also took samples in separate bottles from us and sent those to Lancaster Laboratories and Louisiana State University for analysis, and gave us that data. And I always have to tell people this, they did not give me the analyzed data. I got the raw data back from Lancaster Labs. And so we used the same analysis then to calculate the average, for all the alkanes, the average half-life. We also did the standard technique of taking water from the plume area and then adding marine peptone media. So peptone as the carbon source, that's a standard thing to use, and a marine media, which is a minimal salts media. And going through a couple of iterations of that, getting the cell densities up to 10 to the 5th to 10 to the 6th cells, incubating that at 5 degrees, and then replacing the peptone with Macondo oil. And so this is our mixed consortia. We also did a respirometer study where we actually took water from outside of the plume, not within the plume, and added the Macondo oil directly to that, and incubated it and measured how fast things went away. 4 different techniques, and basically the half-life was 1.2 to 6.1 days. So people thought that was amazingly fast, but it isn't that fast. There's a lot of literature values that show the same thing. And in fact, we did do a ratio of C26 to C15, C26 to C16, C26 to C17, so the shorter change will be degraded faster, so you see a ratio change, and that's exactly what we saw when we looked from 1 kilometer, 5 kilometers, 10 kilometers versus some of these sites, either Macondo oil or directly adjacent to the well. So, half-life comparisons. This is our study in both in situ disappearance and microcosm studies, 4 to 5 degrees, 1.2 to 6.1 days, and with the microcosms, 2.2 to 3.5 days. Actually, amazingly close. However, Al Venosa and Holder had a study in 2007 using C media and the standard enriched consortia technique with crude oil bay oil, at 5 degrees, they got alkane half-lives of 4.5 days and 9.9 days. Again, pretty fast. pH loss from some studies done in the St. Lawrence Seaway, and they went down to freezing temperatures to 5.5. They got 1.5 to 1.7 days pH half-lives. And then also for a DMPA component in the oil, 2.4 to 7.5 day half-lives. And then there's a Chinese study that came out in 2009, and I assume that this G Ming is probably a takeoff on the Corexit. But they were incubating at 10 degrees and got a loss of, or a half-life of 1.6 days for total petroleum hydrocarbons. However, light crude, Louisiana light crude, in soil columns from marsh sediment at 22 degrees degrades much, much more slowly, 11.9 to 69 day half-lives. Lower amounts of oxygen, lower amounts of nutrients, less mixing. It degrades much more slowly if it gets into the sediments, much more slowly. From July 27th to August 26th, we took 170 samples looking for that doggone plume. Remember, this is about 2 to 3 weeks after the well had been capped. All of these samples that you see, these green dots and the white dots, were where we sampled and we didn't detect anything. And on their CTD, we could detect fluorescence levels down to less than 1 part per million. And we couldn't detect anything. But we took samples anyway, brought them back to the laboratory at the depth that they should have been at, and then did a chemical analysis, and for that we can detect down to less than 2 parts per billion. We didn't detect anything, okay, on any of those samples. So and the white dots represent DO dips. So there was apparently still some biological activity here, and it will take the bugs a long time to go back to their natural densities. So, because they go through a cryptic growth phenomenon. So once the oil has gone away and the carbon source has gone away, then the dead bugs, the bugs that start dying, become fertilizer for the ones that remain living. And so it takes a while for this totally to go down. And that's exactly what we see. Now, in a paper that came out just here in January 6, 2011, and they beat me to it, unfortunately, because we had the same data, looking at methane biodegradation. Kessler and Valentine, in their paper, showed that the average methane concentration went down dramatically after the well was capped, but not too much until then, and the oxygen anomaly went up and then came dramatically down. By the end of September, we couldn't detect any methane, and the DO anomaly went slowly away as the bacteria started to degrade. We also did stable isotopic analysis, and when we could detect methane out here in the latter part of July and in early August, we saw that it was highly fractionated. And it was fractionated in such a way that it was biofractionated. So the bugs will actually fractionate it, specifically the carbon that's in the methane. And we knew that had been, the methane that was remaining had been very biodegraded. What they have agrees perfectly with what we had. So there was a lot of reporters that actually went back to us. Now, we have done some other studies with Corexit and oil combinations. And a lot of people ask, well, what happened to the Corexit and how toxic was it? We could never detect the Corexit, even at the wellhead. And the reason for that is that the Corexit is water soluble. That's the reason we use it. And it was being injected at 10 gallons per minute. But at the wellhead, but it dilutes so fast that, in fact, we couldn't detect it at all at the concentrations that we can detect it at. There are components, though, in the Corexit that degrade quite slowly. One of the dominant components of the Corexit is propylene glycol, though. Some of these other compounds can be quite toxic, but they're fairly low concentrations. The propylene glycol is one of the major components. And so what I'd like to tell the public is that those of us that are a little bit older and have had colonoscopies have had propylene glycol the night before. And we can tell you it's a really effective dispersant, okay? And we would hope that the medical community would not be giving that to us if it's toxic, okay? So, and it's funny, that's the longest lived compound. So it degrades very, very slowly. This is at 4 degrees and at 25 degrees. And so it's much slower degradation of those compounds than in the oil itself. And so there could be long-term effects because some of these compounds could be endocrine disruptors, and they could be effective at parts per trillion concentrations. So there's going to have to be some long-term studies done on this. We detected a lot of flocks that were remaining here in late July and August and out in September. And when we detected did sediment sampling. In fact, we did sediment sampling and analyzed the flocks that were associated with the close to the wellhead because we thought we saw some oil there. On almost all the sediment samples, we could not see any visible oil of the cores that we took, and we took 150 cores. And I'll show you the map for that. We haven't completely finished all the analysis of that. There was some oil there, but some of that may have been due to natural seeps. But in the flocks, we could see that there was small amounts of alkanes there, a lot of clays, a lot of carbonates, sulfates, orthosilicates, and aluminates. This is all drilling mud. So remember when they injected for the top kill, they injected all of this drilling mud, that combined with the oil and sedimented out. So we think that's why we're seeing some oil around the wellhead. And we can see some visible oil around the wellhead. In retrospect, that probably wasn't a great idea, but since it didn't work especially. A lot of people ask about the dead zones. And this is from a paper that just came out since this September. And it's from 2005, but the Macondo well was actually up here, and it was flowing in this direction. So it wouldn't intercept this area here. So the dead zones are from nutrients and stuff flushing from the Mississippi River and around, and it's from the marshes and that sort of thing, and the agricultural activities that are associated with this part of Louisiana. And you can see here later on in the summer where they actually had significant drop or a dead zone that would have killed fish at that low DO. We never saw anything that ever got close to that in terms of 2 ppm. The Gulf of Mexico has more natural seeps than almost any other area in the world. It has the equivalent to 400 to 500,000 barrels of oil going into the Gulf of Mexico every year. So the bugs are adapted to it. And it's the only carbon source they have down there. And this has been going on for millions of years. Unlike one of the other areas in North America that has a significant amount of natural seeps is, of course, right off Santa Barbara. And you know, if you've been down there from Los Angeles to Santa Barbara, that in fact you do get tar balls on your feet and things like that. Notice there's no natural seeps up here in Alaska, and in these areas here, what effect that might have in terms of whether there will be bugs that could take care of this if there's a blowout and that sort of thing, we don't know. but it could prevent things from going as fast as what we saw here. So these are the images that are going to be pretty hard for people to forget. This one image here has been hit over 8 million times. But let's use some statistics a little bit. And certainly 11 men were killed here, and I think this is one of the few oil spill accidents where there was a lot of men killed. And as opposed to, there was one bad one in the North Sea. But, you know, it's interesting culturally, we probably didn't give enough attention to these 11 men and their families because it's an international community. It's not a cohesive local community. And so probably not enough thought was given to their families and that sort of thing. But 6,000 birds were killed, and 605 sea turtles, and 97 marine mammals. 4.9 million barrels. And this is according to the government estimates as of October 14th. The Casco Busan spill, that little tiny spill, and this is in millions of barrels, that we had just 3 years ago, killed 2,500 birds. Exxon Valdez spill, 250,000 birds, 22 killer whales, 2,800 otters, and it was only 0.26 million barrels. Oil is all local in terms of its effect. The environment, how fast it's going to degrade, the type of oil it is, whether there's adapted microbial communities, a variety of things like that. On the Moco Cadiz spill off the Normandy coast, in France, 1.6 million barrels, about 20,000 birds were killed, but we don't have very good estimates. On the Ixtac 1 blowout, about 10,000 birds. This was in the Bay of Campeche and is actually the most similar one to the Deepwater Horizon. Houston Chronicle has captured us. They picked up on this. A huge oil-eating microbe just ate Earl. So I've given over 500 interviews on this since it came out in August. And there's a AAAS press conference on Saturday morning, this Saturday morning, and I'm giving one of the keynote addresses there too. So it's not died down yet. We have a variety of things that we're doing in terms of looking at kinetics and oil droplet size. We're looking at respirometer things, and we have papers about to be submitted or submitted. Beach sand, corexit degradation studies, metagenome and single cell sequencing, and I'll talk about this at the keynote address at the JGI users meeting. And all this metagenome and single cell sequencing, there's some really neat stuff here that we did. Surface plume biodegradation and community structure. Did a lot of repeat sampling, and we're just about finished with that. We're doing in situ hydrocarbon analyses and mixing in the deep for analysis. Biotraps, and I'll show you some pictures of that. This is a new technique that we think will be much better for estimating biodegradation rates. Determine kinetics and rates of biodegradation at high pressure. We've set up for that and about finished that. Sediment sampling, we're finishing all that sediment microbial community analysis. It's going to be completely different because it's under anaerobic conditions. Natural oil seeps. BP talked to me yesterday about doing a larger project on natural oil seeps to see how it compares to the MC252. And then long-term marsh studies, we've got those planned, but nothing new yet. Here just shows some of the beach studies that we did and the folks out there sampling. And this shows our bug traps, and these are Cephadex amended beads. DuPont aramid polymer with 75% powdered activated carbon, and they've been fumed with Macondo oil. put into this tube, and then you can see all these tubes along here. And so we got the ROVs. They didn't have anything else to do after the well was closed, so I got them to put out all of these along the riser that was right at the wellhead. And so we put them at 1,500 meters, 1,100 meters, where the plume was, and then at the bottom, and then also at 500 meters where we knew that there's a lot of methane degradation and methane melting occurring, and at the surface. And then they went back and retrieved these at 1 week, 2 weeks, and then 4 weeks, and then the well was declared dead, and they had to demobilize. I was hoping to go out 8 weeks, but never got that far. But you see, this is 4,955 feet, and we got some great pictures from the ROV of them collecting this and that sort of thing. We're just about done with that analysis too. A lot of activity there, a lot of change in microbial structure, and we can see how fast at these different depths the Macondo oil actually degrades. These are all the sediment samples that we took, and we're finishing up all those, and we've gone very far out, also sampled around areas that we know that there's natural seeps, so we can compare to that. And we've gone out in all directions for looking for the sediment analysis too. And that's all, and I'd be happy to answer any questions. Yes?
There's a claim that the person in Georgia who has also been in the press a lot claims there's, as I understand, a couple of inches of sludge on the bottom of the ocean floor. Is that true?
Yeah, I'm going to be in a press conference with her on Saturday morning. That'll be interesting. So it's difficult for us to tell, and I haven't seen any data that she's published on this. She published a paper yesterday in Nature Geosciences. And basically it was from her studies done on the 1st of May, and said there was a lot of methane out there. Well, we knew that. And they did some calculations that the methane should last for a long time and biodegradation rates would be slow and that sort of thing. From Kessler's study, we already know that it's gone too, and from ours too. It's totally gone. And the good thing about the methanotropes, methane oxidizing bacteria, and this is something I have a process patent on with DOE, and it was patented, I patented it over 20 years ago. And it's been licensed by about 30 companies. Methanotropes, the methane monooxygenase and methanotropes degrade about 300 other compounds. So they will deep clean, especially once the methane has gone away and there's no competitive inhibition with the enzyme. They will deep clean it down to less than parts per trillion concentrations. So I think that's an added benefit that we got from the methane being there. But the methane did not start to degrade, and we can show this too, until the oil had basically gone away. Because the oil was too good a carbon and energy source for the bugs, and they weren't going to go to methane until they had to. Or the methanotropes were being outcompeted at that point. But we don't have any evidence. She was seeing some flocks and slime and that sort of thing, and it was just from the back of the boat is what we saw. So I don't know what to make of that yet.
I think her claim is that sludge that she thinks, I think she says is very extensive.
Yeah.
It's basically killing everything on the ocean floor.
She said that initially, but the latest thing that she hasn't said that lately. So I think she's changed her tune a little bit from the latest blogs. I watch her blog now and then to see what she's saying, because she's always interesting. But.
So you mentioned that the bacteria are using some of the nutrients in the water column, the nitrogen
Yes.
and the phosphorus.
Right.
And were you able to measure any difference in the distribution of those nutrients over the depth of the water column? And
yeah.
Also, do you think that any changes might affect the availability of those nutrients in the sort of natural non-oil ecosystem?
No, it's mixing so rapidly, I don't think it'll affect that. But we you know, in the water column, we did not, just in the plume layer is the only place that we saw any depression of those nitrates or phosphates or dissolved oxygen.
So I have, so I was just wondering, so there's been, you know, a really big spill there before, the big stock.
The Exstock one, yeah.
Yeah, in
Yeah.
the 70s. I mean,
Yeah.
on a comparable scale.
Yep.
Known obviously for a long time, there's lots of oil that seeps out in the Gulf of Mexico. How
Right.
surprised were you at the results you got?
Not at all.
So because there's so much hysteria
I know.
in Florida, and I told people, I said, you know, this is really warm water, big oil spill happened
Yeah.
here before, nobody even knows about it.
Yeah, they didn't even remember it, and within 2 years we couldn't find any effect from the Ixtoc 1 blowout. I was around back then, and we couldn't find any effect whatsoever. So let me tell you that ABC Nightly News with Diane Sawyer interviewed me. They wanted to do a story on this back in May, and I told them then that, you know, this is an ecological disaster. I don't want to diminish that at all, okay? And certainly there will be some long-term effects that we need to look at. Is it going to be as dire as the media says? Probably not. At the end of the interview, the producer said, well, how long do you think we'll see the effects of that? It'll be, you know, 30 years or 20 years or 10 years or 5 years? And I said less than 5 years. He didn't like that answer, okay? And he told me for 7 nights in a row that piece was going to be aired, and they never showed it.
So you said that oil degrades quite quickly in the ocean, that's clear. But you said that it takes 12 to 64 days, something like that, half-life in the sediments.
60 days sometimes.
60 days.
60-day half-life, yeah.
Do you know any data about how perhaps oil in marsh sediments has affected the wetlands in Louisiana?
It's hard for us to find effects right now. We can't find it at the surface. But there could be some long-term effects of that oil having sunk into the sediments and also into the sand. We do know it went into the sand and it sunk fairly deep, and that may last for a long time. If the plant roots get down there or animals dig down there, there could be a risk associated with that. But most of that is going to be what we call permanent interment. So it's not going to be a major risk for most animals and plants once it gets deeper and deeper. But that doesn't mean it's a zero risk, okay?
Can you tell us a little bit more about the use formation?
So we thought, you would think that from that color, it looks like iron. We looked at that, it's not iron. And I found some Norwegians, the North Sea has the same type of crude. And they have analyzed it and shown that it's components that were in the original plants that were fossilized to some extent there. It's not clear exactly how that emulsification process works. It's a combination of evaporation and heat, volatile components leaving, that sort of thing, but it does become very recalcitrant. Now the good news is that the tar balls are not particularly, well, you don't eat them, but they're not particularly toxic themselves. But we will see tar balls. Forever, we're going to be, people are going to be finding tar balls and blaming the Macondo well.
What's the role of photooxidation?
Excuse me?
What's the role of photooxidation?
It's probably pretty significant, especially there in the Gulf, because it's very hot and there's a lot of sunlight, and we were fairly far offshore, so there would be a lot of photooxidation before it reached shore. So this stuff was highly weathered by the time it got to shore. The booms, my opinion, I'm not sure they worked real well because of the heavy seas and that sort of thing. I think I would have recommended skimming a lot more and a lot faster and preventing that stuff from reaching shore. Hardest thing there is to do is to tell people that at some point you need to rely upon natural attenuation. In other words, leave it alone. Because any aggressive treatments that you could do in terms of adding fertilizer and nutrients and digging up the sand and mixing it up and it's going to cause way more problems long term. On the Omoko-Cadiz spill, there were areas in the Normandy coast where we couldn't get to. They treated a lot of areas with household detergents and things like that. Within 5 years, in the areas that they didn't treat anything, we could not find any ecological effects. 30 years later, the places that were treated, we still see a significant effect. So the cure can be worse than the disease in this case, in some cases. But there may be situations where if endangered species is going to be killed, then you might want to do some sort of aggressive treatment. You have to weigh these things, carefully.
So you compared this spill to others, saying that for the amount of oil spilled, there was a lot less damage. And I was wondering if that's because of the light oil isn't.
Excuse me?
I was wondering if the reason there was less damage from the spill, given that so much oil was spilled, was due to the nature of the oil?
It's due to the nature of the oil. It's due to the fact that it was far offshore. It was being injected and creating droplets down deep. It's also due to the microbial community being adapted to this particular oil. And there being a very quick response of the microbial community. So there's a variety of things. In fact, Ron Atlas, who was one of the chief scientists, external scientists for Exxon on the Exxon Valdez spill, we are writing a paper comparing the Exxon Valdez spill to the Deepwater Horizon spill, and it'll be in ES&T, I think, next month sometime. So we're finishing that up right now. Because a lot of people have been doing this comparison, and they're really completely different, okay? There was I can go in, there's all sorts of details that a day after the Exxon Valdez spill, a 50-mile-an-hour wind storm hit the spill area. That moved everything to shore. That was devastating. So.